In the last several weeks, we have been discussing B cells, the receptors present on B cells, and the kind of structures present on the cells we know as immunoglobulins, which serve as antigen receptors. We have seen how B cells produce antibodies which are nothing but again the same receptors in soluble form. We have also seen the structure of these immunoglobulins, the classes, the subclasses. We have also seen the functions of immunoglobulins. Now, let us look at the other aspects. Let us look at what these cell surface receptors as well as antibodies recognize. Of course, we know them as antigens. Now, let us look at two words, antigens and immunogens. Antigens and immunogens are sometimes used to describe, not in a very specific manner, what these molecules are. Are there any differences between antigens and immunogens? Both are of course extremely related, but there are subtle differences, and the definition of which I am going to start my lecture with. Immunogen is that molecule that elicits an immune response. It triggers an immune response. For example, in case of the humoral response or the B cells, an immunogen would trigger the cells to proliferate, differentiate, to become a plasma cell that secretes antibody. In case of T cells, it would be that an immunogen would be that molecule that can elicit an immune response in T cells, again for proliferation and subsequently secretion of interleukins or cytokines which help other cells for generating an immune response or for differentiation into cytotoxic T cells. Now, that would be an immunogen, that which elicits an immune response. On the other hand, an antigen would be a molecule that binds to the product of an immune response. Now, in case of B cells, you have guessed it right. The product of the immune response would be antibodies. So, an antigen is that molecule which would bind to the product of the immune response. In case of B cells, antibody, and in case of T cells, it would be that which is able to bind to, like for example, now product of the immune response in this case would be, in case of cytotoxic T cells, would be the target cells. Okay. Of course, there are large number of factors that would be governing immunogenicity. Let us first talk about immunogenicity. What were these factors? Those that would be the properties of the molecule itself, and the second would be contribution of the biological system, that means the host. Properties of immunogenicity with respect to the molecule would mean what kind of a structure does it have, how foreign is it. So, based on that there are at least 4 different properties that confer immunogenicity and which regulate the level of immunogenicity. Now, is there something called level of immunogenicity? Yes, of course. If a molecule is very immunogenic, or we would say a molecule is very immunogenic if it elicits an immune response or antibody secretion which has a high titer and which is also over a long periods of time. So, therefore, that this would mean it is a better immunogen than that which elicits an immune response, but the antibody secretion, the amount of antibody secreted or the affinity and the duration is much lesser. Let us just go over the words. Properties of immunogenicity would be determined by foreignness, molecular size, chemical composition and the ability to be processed and presented with the class of molecules MHC, which probably you are going to listen to from others. Contribution of the biological system, there are 2 that come under this: genotype of the recipient animal and immunogen dosage and route of administration. Now, let us look at this in a little detail. let us start off with the first, that is foreignness of the immunogen. We know already by now that the immune system is based on the central dogma that it has the capacity to recognize self from non-self. So, we know therefore, that the immune response can be activated by molecules that are foreign, that are not present in our bodies, because if you might remember during the development of B cells, those cells which have the capacity to recognize self molecules are deleted from the system, or at least most of them are deleted. A few of them of course continue, but most of them are deleted, which would mean that the first thing, immunogen must be recognized as non-self. Now, let us take an example of serum albumins. Serum albumins, as the name suggests, these are molecules which are almost the same molecular size and present in sera of all mammals. Now, there are of course differences between albumins from one species to another. However, there are also several commonalities. An individual from one species would be able to recognize albumin from another, but because of similarities which would be seen in between species that are closely related, one can say that a host would recognize albumin from the species to which it is not closely related. Therefore, greater the phylogenetic differences between two species, greater is the structural disparity, structural therefore antigenic disparity between them, and therefore, species which are phylogenetically far from each other would definitely be able to mount immune response to one and another's molecules. I have already given you the example of albumin. Let us say now, if I can go with examples, a monkey would not respond to human albumin. However, a monkey would be able to respond by way of course secreting antibodies, would respond to albumin from chicken or from rat. Albumin from rat when injected into mice may not evoke a immune response, because these are closely related rodents. However, a rabbit which is close enough, but yet far away with respect to the immune response, would be able to recognize and mount an immune response to mouse immunoglobulin, mouse albumin. So, therefore, greater the phylogenetic differences between two species, greater would be the immunogenicity. All right. Now, here because I am talking about structure, I have antigenic, okay, because we are talking about the molecule. What we are dealing with right now of course is the immunogenicity. So, foreignness, molecules have to be different from structurally for them to be recognized of course, because self B cells recognizing self or therefore closely related species, molecules from closely related species would be recognized also more or less self. Let us go to the next, molecular size. Immunogenicity is directly proportional to size. This one can understand very easily, because as I have already described to you that if it is a large molecule, a large immunogen, the number of antigenic determinants which are present on that molecule will also be more than a smaller molecule. Let us say a molecule of size 1,50,000 versus a molecule which is only 50,000. There would be at least 3 times the number of antigenic determinants on the larger molecule. So, therefore, the number of B cells that would be triggered to generate an immune response and make antibodies would also be more when we are talking about a larger molecule. So, therefore, of course, immediately immunogenicity is directly proportional to size. One can also look at it from another angle. Larger molecules are retained for a longer period of time in our system, therefore, allowing B cells to recognize and generate an immune response. What is the size of the molecule which can be immunogenic? Now, in earlier times, which would be about 20 years ago, it was thought that any molecule which has a size of more than 5000 or 5 kilodalton has the capacity to generate an immune response. But later on, with more sensitive assays and the ability to even detect small quantities of antibodies generated in an experimental animal, now we know that even small peptides can generate an immune response, provided both, the peptide has both T as well as B cell epitopes. I will of course come to that a little later. So, size is important. The larger the molecule, the better would be the immune response generated. Chemical composition is the third. Now, of course, when I say large size, automatically it would mean that, would it mean that the molecule is diverse or complex in its structure? It need not always be. However, when we come to, we do not think of, we come to natural conditions and not experimental, it holds true. That means, you have an immunogen which is large in size, would definitely have more complexities, more structural variations than a smaller molecule. Experimentally, let us look at whether this would have any relevance. So, what people were, when people were trying to understand immunogenicity, they were looking at synthetic polymers. So, when they generated synthetic polymers of, let us say, simple amino acids, let us say simple one like glycine, they found that if you had glycine chain, a chain of only glycine, the amino acid glycine, then there is absolutely no immune response generated to this. However, synthetic copolymers which have more than 2 to 3 amino acids and in random sequence have some immunogenicity, better immunogenicity than just synthetic polymer of one amino acid. Now, they were able to see as the complexity grows, which I have not written here, they have actually determined the immunogenicity for each of the molecules, but when they had polymers which had more than 2 to 3 amino acids in the sequence, of course, the complexities grew and therefore, the immunogenicity slowly started to increase. Now, interesting here is that when they added to these 2 to 3 amino acids which would be, let us say, hydrophilic, they added tyrosines and phenylalanines at regular intervals. This enhanced the immunogenicity several fold. Now, there is this kind of data available and one can refer to that, which amino acids appear to be harbored in epitopes. Are there such repetitions of amino acids? It is something that one can look at the database and derive, but what we know that if there is complexity with respect to structure, there is better chance of the molecule being an immunogen. What does this ability to be processed and presented with MHC, what does this mean in the context of B cells? You know that B cells recognize through their antigenic receptor or their B cell receptors, which are immunoglobulins, they recognize native conformations. By now, you also already know that T cells on the other hand need to be presented with peptides either in the context of class 1 molecule as in case of cytotoxic T cells, or in the context of class 2 molecules as is the case with the helper T cells. But most B cells, though they recognize native structures, they require T cell help for their proliferation and differentiation. So, most antigens are T cell dependent, that we know, which would mean we also have to think in terms of the immunogen having the capacity to be endocytosed and processed into peptides. Now, why is this relevant? Because after all, one can imagine that antigen presenting cells would definitely have the capacity to also present peptides, but now they have to generate the peptides. So, presentation is one, presenting to T cells is one, but can they generate these peptides? In a regular system, of course, all our cells, all our nucleated cells have proteosomes, and you must have studied this already, which can break peptides into proteins into small peptides, and which can be docked onto class 1 molecule in the endoplasmic reticulum. Similarly, with respect to class 2 molecules, now which are, this is the extrinsic pathway, antigen presenting cells such as macrophages and dendritic cells and B cells have the capacity to endocytose these foreign molecules and now degrade these molecules, and peptides which are generated can be docked onto class 2 molecules for presentation to TH cells. But is there a possibility that some proteins cannot be processed? Yes. Now, in mammals you have amino acids which are made up of the isomer D amino acids. So, therefore, the corresponding proteases would also be able to cleave only D amino acids. Sorry, this I am very sorry, I go back, L amino acids. Now, if we come across peptides, let us say from parasites or plants which have D amino acids, then we will not be able to cleave, we do not have enzymes or proteases that can cleave. Therefore, we will lack of cleavage would mean there would be no peptides generated or very few peptides generated, and thereby they will not be cannot be docked onto class 1 or 2 molecules, and therefore, the immune response is curtailed. Please remember that when I talked about L versus D, we have L rather than D. When we encounter any molecules which have D amino acids, and whether if that would be the cleavage site for a particular protease, it cannot cleave. Therefore, lack of appropriate specificity of proteases would also now make an immunogen less immunogenic, just because T cells cannot be presented with peptides, cannot get activated, cannot therefore differentiate to helper cells or cytotoxic T cells, and if they do not differentiate to helper cells, then B cells will not be able to get those cytokines which they require. Now, again I will repeat what I had talked about in my second or third lecture. B cells require interleukin 4 and 5 for their immediate proliferation after their activation by any antigen or immunogen. Okay. Now we come to the second half. We have talked about the molecule itself, how it can contribute by its properties to be a good immunogen or not. Now, let us talk about genotype of recipient animal. Would the genotype have anything to do with the way the animal responds to an immunogen? Yes, of course, that is very true, because if you just look around and you can see that even in a condition where there is let us say there is an there is infection, you must have heard about these SARS virus, or you must have heard about now the other viruses which the influenza virus, H1N1, and you know that for any people in a locality to get infected would mean that the other people in that locality would have an equal chance to get infected. Now, why did not they get infected? Now, when we say they got infected or not, we always consider the symptoms with which they come, or the symptoms that are they present. A person might get infected, that means the virus would have entered the person's body, but because of a good and well developed immune system, in fact, the symptoms can, the virus is killed, and therefore, there are no symptoms of the disease, which would mean that the immune response in fact is governing the way the infection is settled in or takes root in a particular person. So, let us say 90% or more can mount a good immune response, so that those 90% people or more do not come down with the disease symptoms. They get infected, but they can immediately have a good response, and response which has already been there, or develop a response which can eradicate or completely obliterate that virus. So, therefore, obviously then people with a good immune system or a robust immune system would be able to respond better to any infection. So, the genetic constitution of a host influences the immune response generated. That is obvious from simple observation. But has this been proved experimentally? Yes, there are a large number of experiments that have been carried out, and people have seen that immunologists have tried different strains of mice. Now, there are several inbred strains which are used in experiments, and these are very homogeneous. They are, in fact, they are identical. One strain is identical, all the progeny in one particular strain is identical to each other, and you have another strain which has a completely different haplotype, different MHC molecule. So, now I am just going to discuss with you a very simple experiment that people have carried out. They have taken high responders and low responders. What I mean by high responders? They have already now taken strain of mouse A, strain B, and now they have injected with the same peptide or let us say an immunogen, and then looked at the immune response which is generated by way of antibodies. And they found that some mice even at the same dose to a particular peptide respond by way of large or high titer antibodies, and let us not talk about affinity, high titer antibodies, whereas low responders do respond, but have very little circulating antibodies to that. Now, when this high responder is crossed with low responder, now it was seen immediately that the F1 generation always is intermediate with respect to its response. So, obviously, by Mendelian genetics you know already that genotype of the recipient animal does dictate to a great extent how the host influences the immune response generated. So, MHC gene products. Now, with respect to B cell immune response, you can already immediately say is the helper cells which are required for getting helper T cells required, which will recognize molecules or peptides which are presented to them in the context of class 2 molecules, MHC class 2 molecules, and therefore, the type or the type of MHC gene present would now determine whether the peptides can be docked on or not. And therefore, since now it is the helper cell which are play a key role in modulating the B cell response, MHC gene products play a central role in determining the degree of responsiveness. Now, let us say the low responder one would have the capacity to, let us say, only present 2 peptides, and where the high responder has the capacity to or has the polymorphism at the MHC class 2 molecule. So, that this high responder can now can present 8 peptides. Definitely the 8 peptides presented to that many T cells versus just 2 would after all determine that this particular strain has the capacity to generate a robust immune response. Finally, with respect to what is contributed by the host, the immunogen dose and the route, these two are extremely essential. Is there an optimum dose for any immunogen? That determines immunogenicity. Now, again immediately if you are thinking in terms of an outbreed population of even animals, let us say an outbreed strain of rats, of course, it does not, and therefore, when you come to humans, of course, there is no optimum dose with respect to one immunogen and another. However, if you come down to experimental analysis, then even then optimum dosage differs for each immunogen, because you already know that the complexity of the molecule, the size of the molecule, all these also would play a role on the dosage. If it is a small molecule, less complex, you definitely would require more. If it is a small molecule, it will be thrown out of the immune system, thrown out of the system much faster, will be retained for very short duration, not adequate for the B cells to recognize, and thereby again the dosage needs to be increased. So, optimum dose varies. Very low doses may, well, by the whatever discussion, I mean whatever I said now, there would be no stimulation at all of B cells, because even before the B cells recognize, the antigen is, immunogen is in such low concentration that it is already thrown out of the system. However, it does not mean that an excessive dose is going to alter the responsiveness. If there is excessive immunogen, then B cells enter a non-responsive state. There is too much. There is too much of activation. Let us think in terms of phosphorylation of this molecule. There is just too much. Now, too much of anything is bad. It is bad also for cells. Cells would undergo, B cells which are too activated also, would go into a non-responsive state, will not be able to proliferate. So, very low doses and excessive doses, both of which are difficult to define for any immunogen and has to be done experimentally. I would like to substantiate this with an example, and I find this example really very interesting. Now, this will not be true of most, however, because there is such fantastic variation, I thought I will share this with you. A report which was sometime in the 60s, immunologists were looking at the immune response to a pneumococcal capsular polysaccharide which they used as an immunogen in mice. When they injected mice with 500 micrograms at one time, they found that there was absolutely no antibody response to this pneumococcal polysaccharide. So, they started to decrease the concentration from 50, and when they did it tenfold, they arrived at a concentration of 0.5 micrograms per mouse, and they found there was a good antibody response. Now, in between, I do not remember of course now, but I might guess that 5 micrograms, not 0.5, but 5 micrograms, that is tenfold higher than the lowest dose, did generate an immune response, but 0.5 dictated, in fact generated a better immune response. So, for this pneumococcal capsular polysaccharide, 500 microgram is too much and 0.5 is best. So, you can imagine for every immunogen, in fact, if you want to study the optimum response, one needs to do an experiment. It would be impossible to find this out in any other way. All right. So, now we know what governs or what regulates the amount of antibody generated or the type of antibody generated to an immunogen. You know that it is both host cell derived factors as well as the properties of the molecule itself. Now, we come to the next step. I have been talking about antigenic determinants, and I have been telling you that B cell antigen receptors recognize B cell epitopes. They have to be therefore then defined region on a protein molecule. Now, what are these defined regions? The defined regions would mean that yes, in a given sequence of amino acids, there should be a set of amino acids which would define that antigenic determinant. These are B cell epitopes. So, epitopes corresponding to this B cell receptor, how large are they? Epitopes of native proteins consists of 15 to 22 residues. Now, this can be 15 to 22 residues if it is in a discontinuous array. Now, I will be talking about this discontinuous, continuous versus discontinuous. In case of a sequence, not discontinuous, but continuous array, this number comes down, and it can be, instead of 15 to 22, it can be about 8 to 12. Now, epitope on native proteins consists of this. It would mean that you require that many anyway for a good antigen antibody interaction or receptor immunogen interaction. Now, a smaller subset from the 15 to 22, a smaller subset of 5 to 6 residues contribute to most of the binding energies. but the surrounding, so 5 to 6 are adequate for that actually the core epitope, because of which there will be absolutely no interaction, but the surrounding residues indulge in the complementarity. They also contribute to the forces that govern an antigen antibody interaction, or what is true of antigen antibody would also be true for the B cell receptor immunogen interaction. Now, people have actually determined this to find out the core epitope, and we will come to that later. So, about 15 to 22 amino acids of the antigen and 15 to 22 amino acids of the antibody idiotope. Idiotope is that region at the variable region of the antibody molecule which comes into contact or binds to the antigen determinant, that would be the idiotope. So, the same number of amino acids approximately on the immunogen or antigen now comes into contact with the same number of amino acids on the idiotope of the antibody. Now, I am not going to talk too much about T cell epitopes, because that is something that will be taught to you or discussed with you when you talk about when the T cell and the T cell receptor is presented. But I would just like to tell you what would be the difference between T and B cell epitopes, and therefore, I have to introduce at least a part of the T cell epitope, that there are two distinct interaction sites. Now, this is very different from what you saw in B cell epitopes. There are two distinct interaction sites for T cell epitopes, one for the TCR and other for MHC, because we do know T cell receptors recognize antigens, antigenic peptides to be precise, only when they are docked onto either class 1 or class 2 molecules. Now, are there any differences with respect to the number of amino acids that make a T cell epitope? There is a difference because of the way these molecules need to be docked onto the MHC molecules. In case of class 1 molecules, the T cell the peptide should be about 8 to 11 residues, whereas for class 2 molecules the peptide should be at least 12 to 25 residues. Now, the details of the openness or the groove that is formed by the MHC molecules to house the peptide is different from class 2 and 1 molecule. Just to suffice here to say that class 1 molecule has a docking site which is closed, and therefore cannot accommodate peptides which are longer than 11 residues, whereas the class 2 molecule is a bit open and therefore can accommodate larger peptides. Let us come back to now B cell epitopes. I did say there is something like discontinuous versus continuous. Now, I have drawn a very simple diagram. This does not really tell you which molecule it is, but we know polypeptide chains are have tertiary structures, secondary and tertiary structures. Now, there are epitopes on molecules that are formed by the coming together of regions of that polypeptide chain because of disulfide bond or by the side chain interaction of the side chain of amino acid. Now, in this particular molecule there are two disulfide bonds, in fact there could be more, but I have just depicted two, because of which there are three regions of the same polypeptide chain which come together, and what you see here in red is the epitope. Now, this epitope therefore would be called discontinuous B cell epitope, because on the primary amino acid sequence of the molecule, I mean if you open this out into a chain, a linear chain, then these three would be very far away from each other, but they have come together to form the epitope because of the folding of the molecule. Now, these are discontinuous B cell epitopes. On the other hand, the same molecule which is folded the same way can also have antigenic determinants in a sequence of amino acids in a continuous array. Now, let us look at what these two mean. So let me come with an example. A lot of studies have been done by immunologists or immunochemists on the molecule hen-egg lysozyme. This molecule has 4 disulfide bonds, and of course, I am not depicting all that here. Now, what was an interesting experiment for me to discuss with you is that hen-egg lysozyme, now they were able to identify one region of the molecule which forms in the native structure a loop, and the loop is stabilized by the disulfide bonds, cysteine disulfide bonds. So, what immunologists did was, now they have made the linear peptide which does not have that one of the cysteines, therefore, now this cannot form a loop structure. So, they have had a peptide which is linear, and the same amino acid sequence, now they just introduced a cysteine. In this particular peptide, it would be at the N terminus. So, if you look at the structures, peptide A would be linear and peptide B would be a loop. When antibodies were generated to peptide A and then in another animal to peptide B, then they started looking at the interaction, whether the antibodies generated would be able to recognize cross interact. Now, it was interesting that antibodies that were raised to the peptide A in fact gave a response. There were antibodies that recognized this, but the response was slightly lower. This said that the structure, the complexity in the structure always induces a better immune response. Interestingly, antibodies generated to the peptide 1 recognize peptide 2, but antibodies generated to peptide 2, which is a loop structure, did not recognize peptide 1 at all, because this was linear. What does this mean? That now, if you think in the context of the native molecule, this would represent the native structure and this denatured. Now, if you go back again with the lysozyme, I told you that the hen-egg lysozyme has 4 disulfide bonds. So, if you now break these disulfide bonds, the tertiary structure is gone. The molecule, therefore, it would be denatured, and now if you look at the specificity with respect to the immune response, the antibody generated to the native structure does not recognize, in this particular context, does not recognize at all, but if you think in terms of the entire molecule, would have recognized the denatured form, but to a much lesser degree. Just let us look at, now we say that B cells recognize antigens, so also T cells. You already know one aspect, that is of a difference, that B cells recognize native structures, mostly native whether it is discontinuous or continuous, native antigenic determinants, whereas T cells require the antigen to be processed and presented in the context of class 1 or 2 molecules, therefore, denatured. Are there any other differences? Now, B cells, the receptor is a binary complex of membrane immunogen and the membrane immunoglobulin and the immunogen. T cells, on the other hand, it is a ternary complex, because there is requirement for the TCR to recognize antigen which is which is docked onto class the MHC molecules. B cells interact with soluble antigen, that means when the antigen immunogen enters our body, B cells can immediately recognize, whereas T cells need that step where the soluble antigen is converted to peptides which is docked onto the MHC molecules. Again, obviously, therefore, B cells do not require MHC, whereas T cells require display of peptides by MHC, and this is absolutely essential. B cells recognize proteins, polysaccharides and lipids. So, they recognize proteins essentially. So, proteins would therefore evoke a very strong response, then come polysaccharides and lipids last. T cells mostly proteins. Now, there are of course reports that lipids and glycolipids also can bind to the MHC molecule, not in the way that the peptides bind, but can bind, can occupy that, and can activate certain T cells. B cells, the epitopes are accessible. Why? Again, because we know that B cell receptors recognize native structures, which would mean that in an aqueous phase, epitopes need to be outside on a folded molecule. It should be accessible, because it is only because of the hydrophilic environment, the hydrophilic regions would be hydrophobic patches on the molecule are the ones that are likely to be outside. So, therefore, accessible B cell epitopes can be sequential or non-sequential, as we have discussed, but they need to be outside, and of course, like I have already said, the region should be hydrophilic, should also be flexible. We will come to that in a little while. T cell, on the other hand, the epitopes are linear peptides produced by processing of the antigen and bound to MHC. The interaction between an antigen and antibody depends on non-covalent forces. First, of course, is complementarity, and these between the antigen and antibody, and there are of course large number of structures of complexes of antigen with its antibody, which show that complementarity is what actually drives the antigen antibody interaction, stabilized by ionic bonds, hydrogen bonds, hydrophobic interactions, as well as van der Waals interaction. This is just a pictorial representation. This is the antigen in red, and this is the contour of the antibody, and this just to highlight that there is complementarity. You look at the surfaces of the antibody or the idiotope of the antibody and the epitope of the antigen. Now, this when they come close together, they both would actually have something like a fitting of two, not exactly like lock and key, but similar to that. There is complementarity, and then there are these hydrogen ionic bonds as well as hydrophobic van der Waals interaction. Antigen and antibody interaction are based on the same principle as any bimolecular interaction, any protein-protein interaction for that matter. Antibodies do not alter antigen upon binding. So, it is a reversible reaction. Antigen and antibody forms the complex of antigen antibody, and the forward reaction for forming the complex is K1, and of course, this is a dynamic process and this is reversible, therefore, K minus 1 for the complex dissociating to antigen and antibody separately. So, what are the small K1 and K2? They are the forward and reverse rate constants, and K1 K1, a ratio of K1 by K minus 1, there is a small mistake here. This K minus 1 is what I have written as K2. It is the same as K2 and K minus 1 are the same. So, I should have either had K minus 1 here or I should have put K2 here. So, K1 ratio of K1 by K2 or ratio of K1 by K minus 1 determines the association constant. Now, in how do you discuss this? That if you have an antibody which is very high affinity, then K1 would be stronger than K minus 1. Association would be more than K minus 1, the dissociation. So, naturally, the K1 by K2 is what is going to tell you that the interaction is very tight. Can one calculate this affinity, which is the Ka? Yes, one can. One can calculate by measuring the complex, the molar concentrations of the complex by free antigen and antibody. So, it is governed by the Ka or the equilibrium constant, or the affinity is equal to the concentration of the antigen and antibody complex divided by the concentrations of the antigen and antibody which are free from each other. Of course, needless to say here, the values for Ka vary for different antigen antibody complexes. Now, this becomes even easier to describe in terms of monoclonal antibodies, because now one can generate polyclonal antibodies and monoclonal antibodies to the same antigen, And if one measures the affinity of the monoclonal antibodies, then you can have affinities in the range of 10 to the power 10 moles per liter versus 10 to the power 6 moles per liter. So, a difference of 10,000 or even more. So, affinity is very easy to define. It is the strength of the antigen antibody interaction. So, a higher the affinity, more will be, tighter will be the binding. What is avidity? You may have come across the word avidity. Is it different from affinity? Yes. Now, when one describes avidity, it will mostly pertain to polyclonal antibodies, because in case of monoclonal antibody, as is shown here, a monoclonal antibody, especially to a globular protein, will be able to recognize and bind to two separate identical antigenic determinants, because both arms of the immunoglobulin would be, the heavy and light chain are identical. So, both the arms will bind the same epitope. Now, in the polyclonal, in the context of a polyclonal response, let us say that the antigen which is shown here in yellow, each molecule can recognize 3 at least of different antibodies, which would mean recognizing 3 different antigenic determinants. Now, it is because antibodies are bivalent, and if the antigen is more than bivalent, then you can have a complex formation like this. Anyway, that is not what I am here to say. What I would like to describe is the avidity. So, what has been seen is binding of one antibody to one particular epitope on the antigen can bring about a conformational change in the antigenic molecule, which may allow the binding of the second one, or it may also abrogate, but it can allow, and in fact, it has also been shown that binding of one antibody can bring about a conformation in such a way that the second antibody to the second epitope binds with a better affinity, better, because the conformational alteration has allowed now the antigen antibody interaction, the second one, to be tighter. Now, so a larger number of molecules binding to one, or antibody molecules binding to one antigen, would determine the avidity of the molecule, and therefore, avidity of a polyclonal antiserum can actually enhance or will enhance the affinity binding to a much greater extent than ever achieved by a single monoclonal, no matter how high the affinity is. Now, we talked about B cell epitopes. Can one identify B cell epitopes? Yes, there are of course large number of algorithms that are available now that can predict B and T cell epitopes. But how were these algorithms generated? They had to be generated on certain parameters. So, by way of description of these B cell epitopes, these algorithms were generated, which dependent on whether a region is hydrophilic or hydrophobic. Like I told you, when we are talking about antigen antibody interaction, we are always talking about aqueous phases, which would mean that the most hydrophilic region, or let me put it the other way around, that an antigenic determinant has to be hydrophilic in nature. Only then can it be available for B cell receptor interaction. For them to be hydrophilic, accessibility of hydrophilic, there should also be surface probability. Now, like when I started off saying that hydrophilic, actually there could be other regions of the molecule that are hydrophilic, but yet not a part of the antigenic determinant, because they are not surface exposed. When a protein is getting folded, then of course, there is a possibility that you can have hydrophilic regions which happen to be in the inside of the molecule just because of the folding and nothing else. So, therefore, you also need surface probability, which is accessibility, acrophilicity and flexibility. So, surface probability one can predict, acrophilicity is again being accessible, flexibility. Now, why is flexibility important? We have always been talking about molecules undergoing conformational change. It has been proven by experimental conditions that an antigenic determinant on a protein molecule can undergo changes, slight conformational change, which allows the B cell receptor or the antibody to bind better. So, when kinetics of this binding was looked at, it was found that though the affinity with which the first contact was made, after stabilization or allowing the reaction to become stable, it was seen that same interaction actually resulted in a higher affinity, which would mean that this can only happen when there is flexibility. So, therefore, now all these 4 parameters go to make a B cell epitope. Now, since we talked about all this hydrophilicity, surface probability, etc., epitopes would therefore, B cell epitopes again, epitopes would therefore likely be on beta bends, loops, and not on beta sheets, because usually in all molecules, on protein molecules, the beta sheets are stacked inside the molecule, are internal, and it is the loops that join these beta sheets, and the determinants would be on the loops. Can one identify B cell epitopes? But experimentally, yes, one can do several experiments, but I would like to mention here that identifying B cell epitopes is easily done experimentally, but they would identify mostly sequential epitopes, because for all these methods it is difficult to have the secondary and the tertiary folding or sequences structures, because all these, for example, now limited proteolysis of antigens, solid phase peptide synthesis, all of them would only talk about linear epitopes, but let us go to them anyway. So, that is the identifying B cell epitopes. It is not so easy to identify a discontinuous one. The only way one can identify the epitope is when one is able to determine the structure of an antigen with its cognate antibody by crystal structure. So, any other experiments, it is not easy. However, one can at least identify, map the epitopes present on by these methods. Epitope analysis can be carried out by cleaving, one of the methods is cleaving the protein antigen into peptides by enzymatic or chemical methods. And now you can, let me just tell you the specificity, you can use chemicals such as formic acid, which cleaves very specifically a DP bond, that is aspartic acid proline bond. Not all proteins have this, of course, but if your protein has, you can do that. So, you know already where the protein has been cleaved. So, you generate those fragments. Trypsin cleaves at lysines and also arginines. Clostripane cleaves only at arginines. Cyanogen bromide cleaves after methionine. So, you can look at the amino acid sequence of your protein and you can cleave, now make smaller fragments, and the digested material is then electrophoresed. You can electrophorease these. This is a gel with the digested antigen, and this is the undigested one, and then one can do a Western blot, immunoblot, and then see which are the fragments that get picked up. Now, if it is a monoclonal antibody, you will have very few fragments that get picked up, as compared to polyclonal, you will get almost all the fragments picked up, because there would be that many antigenic determinants. However, now one can then sequence the smallest fragment, and the primary amino acid sequence of your protein. So, which is this particular fragment which would be smallest, this could be around 1 kilodalton, and so at least you narrowed down to the region which has the epitope corresponding easily here, I can say, with respect to monoclonal antibody. Polyclonal would still be a little bit more difficult, but it can be done. Now, solid phase peptide synthesis can narrow down that region further by taking, making small peptides corresponding to that region which was identified, let us say by the earlier experiment of fragment generation, and then you have the amino acid sequence. You can make, in this particular case I have written 13 more peptides were generated or made on solid phase on pins, and which are in each of these wells, and there is a frame shift of every amino acid. So, therefore, now you have peptide 1 which is dissimilar, which is identical to peptide 2 except for the first amino acid, and this one has at the C terminus another amino acid which is from the sequence itself. Now, one can look at the binding of antibodies and then decide. One can do the same thing at the with molecular biology approaches. If for that, of course, you need to have the gene corresponding to your protein, and then you can make overlapping fragments. This is the gene 5 prime to 3 prime, and you can make overlapping fragments sequentially like this, or you can truncate the gene either from the N terminus or the C terminus, and now express each one of them and check for binding to antibodies. And then you know where likely, and by several such experiments, one can now say that this is likely to be the epitope, and later on to peptide generation if you really want to know the co-sequences. Now, why would you want to identify B cell epitopes? What is the reason? There can be molecules such as HIV, which are very one would not want to work with viruses in the lab, and only if you have P3 facilities and in fact much more robust containment facility where you can work with viruses. However, one can, if one is able to identify epitopic regions, then you could be dealing with only proteins and peptides, and therefore, you can go ahead and now design peptide vaccine, which is going to be another lecture. And there are pros and cons of identifying peptides as vaccines, but again that is a matter for a discussion which is related, but not identical to this. So, now let me summarize. Immunogens and antigens are similar, they are relevant of course. All immunogens are antigens, however, not all antigens are immunogens. B cell epitopes and T cell epitopes can be identified based on whatever I have discussed so far, and one can use these for vaccine design. Now, let me just tell you why all immunogens are antigens, but the reverse is not true, and I will give you an example. You have now haptens, which I have not even begun discussing, and I will do that when we deal with vaccine design, peptide vaccine design. Haptens are small molecules. They can be antigens, but they cannot be immunogen unless they are conjugated to a carrier protein and then injected. The antibodies that are generated will be to the hapten as well as to the carrier protein. So, therefore, now it will be, and in fact, the antibodies will also recognize the hapten alone, not only as conjugated with the carrier protein. Therefore, we know that haptens can be antigens, but not immunogens. They can of course be made immunogenic by conjugation. So, we will be dealing with the vaccine design and what are the pros and cons in a subsequent lecture. Thank you.